Hello everyone and welcome to the AMDA webinar. Your speaker for today's program is Andrea Dardis, Head of the Lab at Regional Center for Rare Diseases at the Academic Hospital of Udine. Audiocast quality is subject to your equipment, available bandwidth, and internet traffic. If you experience unsatisfactory audio quality, please use the telephone dial-in option provided in your confirmation and reminder emails. If you have dialed in, operator assistance is available by pressing 0#. You can send questions at any time by using the Q&A window located on the lower left of the presentation screen. Just type your question at the bottom of the Q&A box and click on the Ask button with your mouse to send it. A question and answer session will follow the presentation. Now I will turn the call over to Andrea Dardis. Please begin, Andrea.
Okay, thank you very much. First of all, I would like to apologize because I was not able to attend the conference in San Antonio, but I would like also to thank AMDA for giving me the opportunity to present our work in any case today. So I will talk today about the project that we started in Italy many years ago, like 10 years ago. And this is a collaborative project between our group in Udine and the group of Dr. Emanuele Buratti at the International Center for Genetic Engineering and Biotechnology in Trieste. So, as I think everybody knows in this audience, Pompe disease is caused by deficiency of the alpha-glucosidase activity, and this enzyme, the lysosomal enzyme, is encoded by the GAA gene. So this gene is located in chromosome 17 and has 20 exons. The first ATG is located in exon 2. So today, more than 500 mutations have been described in this gene to cause Pompe disease. And most of these mutations are private or present in a very small number of families. Probably the only exception is this mutation, the C-32-13 which is generally called IDS1 mutation, which is present in about 40 to 70% of alleles in patients affected by the late onset form of the disease. So since this mutation is quite frequent, it's very interesting, and we started to work in this mutation, as I said before, many years ago. So the mutation was described like 20 years ago by the group of Nina Raven, and this is what is called a splicing mutation. So for those who are not familiar with the splicing process, what we have to keep in mind is that for a gene has the information to synthesize a protein. So this gene needs to be first transcribed into a messenger RNA, and then this messenger RNA is translated in a protein. So for this messenger RNA to be translated in a functional protein, this mRNA has to be processed by what is called the splicing process. And in this process, what happens is that some sequences called introns are excluded from this messenger, and all the sequences called exons, which are here, are stick together. So this mutation, the IDS1 mutation, is located within the intron 1 of GAA. So the effect of this mutation that was described by Nina Raven group, it's that in the presence of the mutation, there is a decrease in the efficiency of this splicing process. So if we analyze the messenger RNA, which I will call mRNA from now on, in a control, in a healthy subject, what we found is that most of this mRNA is normally processed, and it's a normal, in this normal RNA, exon 1, 2, and 3 are stick together. But still, there is a small portion of this messenger that is not correctly processed. and the exon 2 of GAA is partially or totally excluded. So what happens in patients carrying the IDS1 mutation is that this deficiency of this splicing process is significantly decreased. So in the patients, what happens is that most of the messenger RNA is processed in an incorrect way. So, but still in the patients there is some normally processed mRNA. So in the patients, still there is a small portion of normal messenger. And these patients, a small portion of normal messenger can be translated in a functional protein. So that's why these patients present a residual activity and present the late onset form of the disease. So this mutation is a very interesting target for therapy for two main reasons. On one hand, almost all late onset patients carrying this mutation in at least one allele. And on the other hand, these patients express some residual GAA activity. So, I don't know what happened. Okay. So, and some of these patients express up to 25% of normal GAA activity. So just a little increase of the exon inclusion, so the efficiency of the splicing, could be enough to achieve a beneficial effect in clinical settings. So how we could increase this efficiency of the splicing? So in our project, we try different approaches to try to increase the production of normal splice mRNA, which would be translated in an increase of residual activity. So as I said before, in a normal condition, all the intronic sequences are removed and exon 1, 2, and 3 of GAA are stick together. So for this to be efficient, it is needed the presence of sequences within the mRNA that are called enhancer, sequences that bind factors that positively increase the efficiency of the splicing, and there are some sequences that are called silencers that bind factors that decrease the efficiency of the splicing. And the splicing to be corrected, we need a balance between these factors. So what happened in the case of the IDS1 mutation is that the mutation almost abrogates the binding of a positive factor, which is called U2AF65. In this condition, there is an imbalance between positive and negative factors, and there is a decrease in the efficiency of the splicing. And for this reason, exon 2 is completely or partially excluded from the messenger. And this causes since the first ATG is located in exon 2, no protein is translated. But remember that these patients still a little bit of correctly spliced mRNA is produced. So one approach we tried first was we thought that if we could block the silencer sequence, so we could abrogate the binding to the mRNA of a negative factor, we could somehow reestablish the equilibrium and the balance between positive and negative factors. So one way to block a silencer sequence is to use the antisense oligonucleotides, which are sequences that bind this portion of the mRNA and abrogate the binding of the negative factor to the messenger. So to do so, we first needed to identify these silencer sequences. So what we did first was to construct a series of deletion contracts in which we excluded from the exon 2 some portions of the sequence. So as you can see here, in the case of the mutant construct, we almost do not produce any normal mRNA, which is here. So, but when we delete this particular region, we got an increase in the exon 2 inclusion. Remember that to synthesize a normal mRNA, we aim to include the exon 2 into the mRNA. So, at this point, we thought, okay, this region could be our target for the design of these antisense oligonucleotides. So, indeed, we designed a mixture of three antisense oligonucleotides targeting this region, and we treat fibroblasts from a patient carrying this mutation in one allele, and we can see that indeed, when we treat the cells with this mixture, we were able to increase the expression of normal RNA, and what is more important, this increase in the expression of normal mRNA caused an increase of about 50% of GAA activity. So we then tried the same approach, but using now myotubes from a patient carrying this mutation, and we got almost the same effect. So we were able to increase the inclusion of exon 2, and we were able to increase about 40% GAA activity, and what is more important, we were able also to decrease the accumulation of glycogen within these myotubes. So this is a possible therapeutic approach that in vitro is working quite well. So another approach we wanted to try was the use of what are called small molecules. So the idea is always to restore this balance between positive and negative factors. So another approach could be the use of small molecules, chemicals, that increase the expression or the activity of factors that influence in a positive way the splicing process. So we decided to screen for this kind of molecules. So to perform this screening, we decided to perform what is called a high-throughput screening of molecules able to increase exon 2 inclusion. And in a high-throughput screening, you screen from 100 to 1,000 of molecules. So the first step in this part of the project was to design a system, an in vitro system, that could be used for the screening of such amount of molecules, because this is not possible to do with fibroblasts from patients, for example. So we designed this reporter system, splicing reporter system, in which we clone the exon 2 of the GAA and the intronic flagging region, including the mutation, the IDS1 mutation, in the middle of the coding sequence of GSP. GSP encodes for a fluorescent protein. So in the presence of the mutation, the efficiency of the splicing is decreased. So what happened is that the exon 2 is excluded from the mRNA. So these two portions of the cDNA of the GSP stick together, and this is translated in GSP, which is a fluorescent protein. So when we transfected cells with this construct, we get a green fluorescence. If we found a molecule able to increase the efficiency of splicing and increase the inclusion of exon 2 within the messenger RNA, what happened is that now in this mRNA, there is exon 2 in the middle of the coding sequence of the GSP. So at this point, this cannot produce a fluorescent protein. So the consequence is that there is a decrease of the fluorescence of these cells. So we aim to find molecules that decrease the fluorescence in this reporter system. So we screened different libraries, and the first one was we performed a high-throughput screening of 1,200 molecules that are FDA-approved small molecules. And this approach has the advantage that if we found a molecule able to increase the normal splice mRNA so the inclusion of exon 2 in the mRNA, these molecules are already approved by FDA for treatment of other kind of diseases. So we could repropose, this is the basis for a drug repurposing. So we performed 2 rounds of screening, and we found different molecules, 6 molecules, that were able to decrease the fluorescence in our system. So at this point, we used these molecules to try to treat cells transfected with an imaging carrying the mutation, and we found that among the 6 molecules, only 3 were able to increase exon 2 inclusion, so normal splicing. And when we use the same 3 molecules in patients' fibroblasts carrying this mutation, and we measure GAA activity, we found that only 1 significantly increased the activity of this enzyme. And this molecule is the ferroxamine, which is currently used for the treatment of hemochromatosis, and it's a very well-known iron chelator. So we decided to further validate these results. So we treat fibroblasts carrying this mutation with this molecule, and we found, as there is an example here where you can see that the ferroxamine increased the normal spliced mRNA in fibroblasts and increased about 2 times exon inclusion, and this was associated with about 40 to 50% increase in enzymatic activity. We tried different cell lines. As you can see here, these are other 2 cell lines, both carrying this mutation from different patients in 1 allele, and we got the same effect. There is an increase of 50% of enzymatic activity when they are treated with the ferroxamine. And it's very interesting that we treat a cell line that carry another mutation. It's also a splicing mutation, but it's a different one. And we got the same effect. So it looks like the ferroxamine acts in increasing the efficiency of the splicing in the presence of mutations that affect the exon 2 inclusion. Not only this more common mutation, but also other mutations located in the same region. So since, as I said before, the ferroxamine is it's a very well-known iron chelator, we treat the cells with the ferroxamine fuhemin to overload the cells with iron. And we found that in the presence of hemin, the effect of the ferroxamine was completely reverted, both on exon 2 inclusion and GAA activity. So it looks like this effect of the ferroxamine on exon 2 inclusion and GAA activity is somehow modulated and mediated by the presence of the iron availability in the cells. So on the other, we now are trying to understand a little bit better the mechanism by which the ferroxamine acts on exon 2 inclusion. So it is known, as I said, that the ferroxamine is able to reduce the amount of iron in the cells, since it's a chelator of iron, and in turn is able to increase the activity, increase the abundance in the cells of hypoxia-inducible factor 1, which is a factor that is transcription factor that is induced by hypoxia. So we wanted to see whether this effect on exon 2 inclusion of the ferroxamine is mediated by if1 alpha. So to do so, we treat the cells with an inducer of if1 alpha, which is cobalt chloride, and we measure exon 2 inclusion. So as you can see here, when we treat cells with cobalt chloride, you can see that there is as expected, there is an induction of if1 alpha. But in the case of this treatment, we see that there is no increase in exon 2 inclusion. So this means that the effect of the ferroxamine on exon 2 inclusion is not mediated by if1. But we were a little bit surprised when we observed that although if1 alpha induction of if1 alpha was not able to increase XO2 inclusion, we observed in cells treated with cobalt chloride or DMOG, which is another inducer of if1, we observed an increase in GAA activity. So to further confirm this result, we treat with deferoxamine cells that were silenced for if1. So these cells do not express if1. And what we observed is that in the absence of if1, there is a partial reversion of the effect of deferoxamine on GAA activity. So from this from this experiment, we conclude that deferoxamine is able to increase residual GAA activity, but by two different mechanisms. On one hand, it increased the efficiency of splicing in the presence of the mutation, so there is an increase of normal spliced mRNA, and this is translated in an increase of residual activity. This effect is dependent on iron availability, but is independent of the expression of if1. On the other hand, deferoxamine is able to induce if1 expression, and this somehow increased residual activity by a mechanism which we need to still understand, but it's independent on the exon 2 inclusion. So now we take advantage of our splicing reporter system, and we are screening other libraries. These are very, very preliminary results. We obtained these results just last week, but I wanted to show you that now we screen a phytochemical library of 320 compounds and a fragments of chemicals library of 480 fragments. And we performed 2 rounds of screening of these 2 libraries, and you can see here, this is the correlation between both runs. And we identify, remember that we need to identify chemicals or phytochemicals that are able to decrease the fluorescence of these cells, and we have identified 5 hits in the phytochemical library and 5 hits in the fragment library, and we need to validate this data right now. So this is more or less where we are now with our research. So the future steps, of course, are the validation of these phytochemical and fragment library hits. We want to better characterize the mechanism of action of deferoxamine on GAA, because this could also lead to the identification of new therapeutic targets. So now we are doing RNA sequencing of in cells treated and not treated with deferoxamine to try to understand the mechanism by which deferoxamine acts. And of course, and this is very important, we need to validate the effect of deferoxamine in muscle cells, because as everybody knows, muscle cells are really the target of the disease, and we need to target these cells. So we need to see whether deferoxamine is able to include exon 2, to increase exon 2 inclusion and to increase enzymatic activity in muscle cells, and if this increase is enough to at least partially decrease the glycogen accumulation in this cell. So as I would like to acknowledge people that are working in this project, in particular Emanuele Buratti from the Molecular Pathology Laboratory in Trieste, who is a partner in this project, and he's really an expert in splicing, the process of splicing. The screening has been done at the ICGB by Luca Braga, and all the experiments in the cells were performed by Paolo Peruzzo in my laboratory. And I would like to thank our supporters, Fondazione Teleton, AFM Teleton, Eurobiobank, the Associazione Italiana Glicogenosis, and of course, AMDA. and all of you for your attention, and I will be happy to take questions now.
Thank you, Dr. Dardis. We do have some questions at the bottom of the screen. If you want to submit your questions, all you have to do is just use the Q&A window at the bottom of your screen, left side, and then type your question, and we will receive it. Thank you very much. We have a first question. Our first question is it possible to correlate the amount of correctly spliced SV1 with patient function?
Well, actually, in theory, it should be possible to do that. But really, I mean, the amount of correct splicing could be a little bit different, for example, in fibroblasts than in muscle cells. So it is quite difficult. I mean, although, as I said, in theory, it should be possible, it is quite difficult to really understand the amount of normal spliced mRNA present in the muscle. And on the other hand, there is very nice work done by the group from Rotterdam, where they show that there are also other factors and other, for example, polymorphins and sequences that could modulate and could also be part of this process, and also could modify the amount of correct splicing in the muscle, in the cells. But in theory, I mean, if you have more correctly spliced mRNA, you should be able to produce more protein, and of course, you have to the cells could work better. But there are also other factors that could influence the phenotype. So it's not only that. I don't know if I understand. I mean, I answer your question, Lisa. Okay.
Thank you. Thank you, Dr. Dardis. We have another question from her. For the silencer regions that were attempted, there is a star on lane 8 and a band that doesn't appear to be associated with a splice variant. What is that?
Okay, let me go to, I know what you mean, but just let me check, just a second, please. So I go here, line 8, yes, absolutely. Here, actually, I didn't show, I'm sorry I didn't show because I had a scheme of what is happening, but when we deleted, just a second, I want to show you. So, when we deleted this region here, we found that there is another cryptic splice site that is activated. So the deletion of this region not only, I mean, there is almost, there is an effect on the normal spliced mRNA, so it looks like there is a silencer there. But this band is because when we delete this, there is another splice site that is activated and generates another aberrant spliced variant. So that's why we didn't target this sequence, because it looks like it could work, as I said, because it is an increase of normal spliced mRNA, but at the same time, there is another cryptic site that is activated when we delete this region, so it's not really, I mean, we didn't like that. So that's why we didn't use it. But yeah, you are absolutely right. This band is because there is another site, a cryptic site that is activated.
Thank you, Doctor. And our next question, what is the potential clinical relevance of iron overload in patients that might be treated with Defe? Would therapy be most effective in patients not receiving iron supplementation, for example?
Okay, I'm sorry, but I cannot see the just a second, hold on. I cannot see the question. Why you cannot see the question?
Oh.
Okay, I got it. Okay.
There you go.
Yes. sorry, what is the potential clinical relevance in patients? Okay. So we don't know that. We really don't know that. This is something that is really worrying us, because it's true that deferoxamine, of course, it's effective, and it's when there is an iron overload, for example, in patients with hemochromatosis. But we don't know what would be the effect in patients that do not have iron overload, like the Pompe patients. And we don't know if this, because it's true, maybe the deferoxamine will increase exon 2 inclusion and enzymatic activity, but what happens if the deferoxamine decrease the iron levels in the patient? So this is something that we really need to be very careful. That's why it's not so immediately that you can, go to clinic with this compound. And that's why also we really would like to better understand the mechanism by which the deferoxamine acts, because maybe we could, when you identify all the steps in the mechanism, maybe you can act downstream, or you can act directly on the mechanism that is involved in exon 2 inclusion, increasing exon 2 inclusion, but without the need to move the iron availability in the cell.
Thank you again, doctor. And last question, are you considering evaluating Defe in a Pompe mouse model?
Well, this is the Pompe mouse model. It's a problem, because it looks like, from our data, it looks like the deferoxamine is working specifically on splicing mutation, and the Pompe mouse model it's a knockout mouse. So this treatment would be mutation specific, and the mouse does not have this mutation. So we would need to develop a mouse model with the mutation, specifically this mutation, and then use the deferoxamine to try it. So we are considering to develop this mouse model, and but it's not so easy, because we were doing some experiments using the mouse sequence, and in this region where the mutation is located, the sequence of the mouse gene is extremely different from the human. So to develop a mouse model with this particular mutation will need a little bit of work, because as I said, for example, in this region there is a completely different sequence. So you really would need to knock out this gene in the mouse, and then do a knock-in with the human gene carrying the mutation. So we are considering this, but it's not so. The model available right now is not useful for this kind of work, unfortunately.
Thank you, Dr. Dardis. And that was the last of our questions.
Okay. Thank you very much.
And this concludes today's AMDA webinar. Thank you, everyone, for attending.
Thank you, everyone.
The moderator has ended
Bye.
the conference. Goodbye. Thank you for